All right, we're on SOL Strand number 2, life at the molecular and cellular level. Let's get started. So the above graph shows how dissolved oxygen and CO2 levels changed in a pond over a 24-hour period. What caused the decrease in oxygen concentration during the night? All right, so when is the sun out? Sun's out during the day, right? So if the sun's out during the day, will you see more oxygen in the pond or less? So let's try to understand photosynthesis. Plants take in CO2 and give off oxygen. Let's just write the formula. Oxygen plus glucose. Okay, so as you can see, if the sun is out, you can do photosynthesis, and so you'll take in CO2 and increase oxygen. So that makes sense, right? During the day, we see oxygen being absorbed, CO2, and then we see oxygen increasing. So the answer is, why do we see decreased oxygen during the night? Well, that's because of decreased photosynthesis. During the night, there is no sun, so there is no oxygen. Moving on. The process of photosynthesis and respiration can be thought of as cycles because, they're the opposite of each other. So for photosynthesis, we take in CO2 and we give off oxygen. Plants, yes, hello kitty, I'm a little busy. So we also, let's just write them down. Sunlight, CO2, and then water in the plant pot. And that's going to give you oxygen and glucose. So then, what do humans eat? Well, we have to breathe in oxygen or we die, right? I once heard a funny joke. Someone was like, you have a short time limit on life, but you refresh it every time you breathe. I thought that was funny. Anyway, we breathe in oxygen and we eat things like sugar. And our mitochondria give us ATP. Now, quick disclaimer, plants also have mitochondria, so they actually burn the sugar they make. I mean, why would they make it if they're not going to burn it? Just want you to remember that. So the answer here is the reactants and products are kind of flipped. Plants make glucose and oxygen, and then animals basically take that in to make ATP. So it's just the opposite reactions. That's the circle of life, if you will. Number 3, let's see. The diagram shows the process of osmosis. Only water molecules could enter the cell because of what? So this has to do with a term called selective permeability. So membranes, like a cell membrane, which is what this thing is, they are selectively permeable, meaning they let some things in, and some things they just don't let in. And that can depend on several things. It can depend on size, it can depend on the charge of the molecule, its polarity, all that kind of stuff. In this example, it's just size. So this molecule here is too big, and this one is not, so it goes straight through. Same thing here in this example that you got from your quiz. The circle molecule, which is the protein, it can't get in. As you can see in the before and the after, there's only 3. So you can see 3 proteins. No change in the 3 hours. Whereas the X molecules, which represent water, you can see that they go into the cell within those 3 hours. So it's just size. So let's find that one. Sorry, you can probably hear that truck in the background. That's I'm outside. I like being outside when I record. Have more energy, smaller. Oh, here it is. Yes, the water is smaller, so it can just go through. So that's selectively permeable. Your cell membrane is selectively permeable. Okay, next, the cell theory. So what is the cell theory? It was developed by 3 people. So Schleiden basically said that all plants are made of cells. Schwann said that all animals are made of cells. And then Virchow said that cells come from other cells. So that's the 3 parts. And we've actually expanded upon that in this class. And so here's the actual 6 parts. All living organisms are made of cells. If you are alive, you are made of cells. And that includes single-celled organisms. Next, cells are the basic unit of life, meaning that the stuff inside of them isn't alive, but they're considered alive. Number 3, cells come from pre-existing cells. That's what Virchow came up with. So mitosis, meiosis, and also sperm and egg are cells that make you. Let's see. Heredity. You have DNA inside of your cells. It designs your cells. 5, cells have basic chemical composition. We're talking fats. We're talking, for the cell membrane, we're talking DNA. Let's see. Energy. So we have mitochondria. Plants have chloroplast. We have some sort of metabolism, which means a way in which we process our energy. So those are the parts of the cell theory, and which one of those sounds like it encompasses at least one part. So let me just write out the 3 that the SOL test seems most concerned with. The first is that all living things are made of cells, so made of cells. The next is that cells come from pre-existing cells. And the third one they seem to care about is that they are the basic unit of life, right? So let's see if any of these 3 sound like that. So the first one does. Cells are the building blocks. So that sounds like they are the basic unit, and they are all living things are made of cells. So we're going to go with that one. But let's just rule out the other ones. Cells come in different shapes and sizes. I mean, yes, they do, but that really doesn't check off any of these here. Cells contain a nucleus. Not all cells. This is a bacteria, and as you can see, its DNA here is not contained in a nucleus. It's just sort of stringy, and it's kind of just hanging out in the cytoplasm. They are not big enough for a nucleus or any organelle like that. They're just too small. So that's actually not even true at all. Cells can be seen through a microscope. Yep, that's true, but that is not part of the cell theory. So you just kind of have to know what the cell theory is. Coming up next, we got number 5, and we're moving at a good pace. That's good. Which of these functions most like the brain of a cell? So what does it mean to have a brain? Well, in your brain, you process information, so it's like the information center. So what can we think of that is information in your cells? Well, DNA is. DNA is actually information that is passed down from your parents. It's the information, it's like a cookbook, that designs your cells. Now, all of your cells actually have the same exact DNA. Your fat cells, your liver cells, your heart cells, they have the same exact DNA in all of them, and yet there's 20,000 different types of cells. And so how can that be? Well, there's genes. So you can have gene 1, gene 2, gene 3. You have about 20,000 different genes, and you have about 20,000 different cells. And so what ends up happening is you just read different sets of genes on that DNA for different cells, and you just don't read the others, right? So for fat cells, you read fat cell DNA. For heart cells, you read heart cell DNA. You have every single recipe to make every single cell inside of every cell, but you only read the pertinent information for that cell, and that's called differentiation. So that went a little off topic, I'm sorry, but where is all this stuff held in a eukaryotic cell? It's going to be in the nucleus. So the brain of the cell is the nucleus, because that is where DNA is going to be. If we look at these other ones here, they're important. This is like the UPS, the Golgi apparatus. It sends off little vesicles filled with stuff, either to other cells or to the cell itself. Then we have the mitochondria, which generates ATP from the food that you eat. And we have the smooth endoplasmic reticulum, which sort of comes off here, and it's going to help make the cell membrane. Also hormones and fats. All of them have a different part, a different function. Okie doke. So in cows, long hair is dominant to short hair. I like to keep notes on this just to keep track. Long hair is dominant, and that would mean short hair is recessive. I do hope on your SOL that you do take notes like this. And so it's going to talk about a heterozygote. So I'm using T's here, so I'll just use T's. If it's a heterozygote, it's going to be a capital letter and a lowercase letter. Hetero means different, right? And so if you have a heterozygous genotype, that means you have one of each allele. So we have a tall allele and a short allele, and that just means that we're going to be tall because the dominant allele wins. All right, so now it's asking what percent of the cells undergoing meiosis will carry the dominant allele. So this is pretty simple. You know that you are only half of each parent, right? You're half your mom and you're half your dad. Likewise, when you have a child, that child will only be half of you. Therefore, it should make sense that you only give half of yourself. So how do you only give half of yourself? The answer comes in when you go through meiosis. When you're making that egg or you're making that sperm, you are actually only giving half of your DNA. And the way that we do that is a process called segregation. During meiosis 1, remember there's 2 phases of meiosis, meiosis 1 and meiosis 2, where we actually split up our homologs, our homologous chromosomes. Homologous chromosomes come from your parents, and of course you have one of each from each parent, and you're going to actually split those apart randomly. And so when you do that, you're going to separate alleles. And so half of them will go this way and half of them will go that way, and your child will randomly get one or the other. And so that contributes to the overall randomness of reproduction and making your child so unique. And so in this example here with this heterozygote, you could pass on this allele or you could pass on this allele. And so that's going to be a 50-50 shot for that child. All right, 7. People who have been exposed to excessive radiation often experience mutations. If these mutations only occur in somatic body cells, these people may what? So we have to go over this in layers, and the first thing we have to talk about is mutations. And so there's 2 types of mutations. There's point mutations and chromosomal. Point mutations are, if this would just work, point mutations are when you change only a single letter in DNA. And so what do I mean by these letters? Well, if we look at a sequence of DNA, it has random sequences of A's, T's, C's, and G's. So in a point mutation, you are changing these individual letters. And so you could delete a letter. You could add a letter. You could substitute a letter. How do I do that? I have to erase one here. Let's get rid of that T, and let's put in something like a G. Oops, come on. What you doing? What you doing? Oh. So those are the 3 different types. We could delete, we could add, or we could substitute a letter, and that's going to change everything for you. So for example, my brother, he has cystic fibrosis, and that is a substitution. And that just means that he has one letter substitute for the other. And so if we remember gene expression, it starts at genes in your DNA, and then you actually make proteins. And those proteins are going to make your traits, right? And so what happens with him is his genes changed from that substitution, and that changed his proteins, which basically makes it so that he can't flow water through his cells correctly. He can't undergo meiosis correctly. His cells dry up, and he actually ends up with a lot of problems. And so that has changed his trait, right? Because his protein changed. So anyway, that's a point mutation, and that's just on the single letter level. And then we have entire genes that can change. Now remember, genes are on chromosomes, and they can be thousands of letters long, thousands of nitrogen-based letters long. A's, T's, C's, and G's. And so this is changing an entire gene, right? And so here are some examples. We could have a deletion. So we're just going to delete gene D. So if, for example, gene D was melanin, a pigment in your skin, and we deleted that, that could result in something like albinism. When you have those really, really pale kids with red eyes and like white hair, white skin, they have absolutely no melanin because the gene has been lost. Okay, so then the next is duplication, where we just take a gene or 2 genes or 3 genes and we just duplicate a whole nother set. So actually, this isn't really that impactful. You have duplications in your genome, guarantee it. It really doesn't hurt you because your cells can turn off the extra set. And actually, duplications are great fuel for evolution because it gives us some DNA that can mutate, and it really doesn't matter if it goes the wrong way, right? So you can still have like a backup gene. So it's a really great way for evolution to occur, and that's one reason why your cells tolerate it so well. Okay, the next, an inversion reverses a segment. So we could inverse genes. So here we have gene B and C reversing with D. And so the reason why that's an issue is genes on chromosomes are a lot like ingredients in a recipe book. You are made of ingredients, right? When you are developing in your womb, in your mom's, some people call it the oven, like you got a bun in the oven if you're pregnant. So it's kind of like that, actually. You sort of are in an oven, and you sort of are being cooked. You're being processed, right? And so just like cooking something, you've got to put the ingredients together in the right order if you want to make the right thing. If you have a cake, for example, and you add flour after you've baked the cake, well, you're just going to end up with an eggy, dense mess of egg and just like chocolate and a bunch of powder on top of it. That's not a cake, right? Same thing with you. If we have your genes in the wrong order on your chromosomes, they're going to fire in the wrong way when making you. And so when we have this type of mutation, it tends to result in basically a stillborn child. The next is a translocation. This also will create sort of a stillborn child, and this is the reason why two members of different species cannot create fertile offspring. Like a lion and a tiger, for example, they can make a liger, which is a hybrid animal, but that liger cannot create children because when it undergoes meiosis and tries to do crossing over with non-homologous chromosomes, they end up switching genes that just make these weird chimeric chromosomes like you see here. And so, for example, if this is the chromosome of a lion and this is the chromosome of a tiger, when they try to go through crossing over, which is when segments of genes switch between parents, they're going to create these weird chromosomes that don't have all of the genes necessary to create a living thing. And this is why two different species cannot create grandchildren together, because their children cannot undergo meiosis correctly. Anyway, so those are the different chromosome mutations and point mutations. And now we have to talk about the cell types, because that's another element of this question. And I'm sorry this is such a long explanation for one question, but it's really an involved question. Okay, so germline mutations versus somatic mutations. So first of all, what is a gametic cell versus a somatic cell? Well, a somatic cell is any cell in your body other than sperm and egg. So it's most of your cells. They are created through a process called mitosis, where we just clone our cells. Next is the gametic cells, which are sperm and egg. And so changing the DNA inside of them has different consequences. So for example, the way I like to think about this is germ cells, like sperm and egg, they are a lot like seeds. And those seeds here will grow into a plant, an individual. They will grow into an adult. And so if you change the seed, you're going to change the entire adult. Every cell that comes from that egg and sperm will also be changed, because again, you're changing the DNA here to by mutating it. So I'm just going to say this little part was mutated, and that's going to make every single cell different. Whereas if you just mutate like a somatic body cell, it's only going to change that portion of the individual. It's not going to be in every cell that comes from that, right? So it's sort of like if you change the seed, you change everything in the plant. Whereas if you just change one cell in a plant, you're not going to change the entire thing, right? So that's basically what that's talking about. And next we have to talk about the cell cycle. So what's a bigger impact, changing a somatic cell or a germ cell? Well, we know that changing a germ cell is a bigger deal. It will change the entire individual. And that's usually like the fuel for evolution. And so this isn't really an evolution question, so we can probably assume that and it even says in the question that they're talking about somatic cells. So what generally happens when you change a body cell is you might get like a mole or a freckle, or even in the worst case scenario, cancer. And so let me explain why you get cancer when you change body cell DNA. The reason why is because you have a lot of segments of genes on your DNA that have, so we'll just say this is a gene here. You have a lot of genes that create what are called cyclins. These cyclins are meant to regulate your cell cycle. And here you can see a picture of a cell cycle. We have M phase, which could be mitosis or meiosis, where cells are doubled. And then we have the normal life cycle of a cell, which is called interphase. And inside of interphase, we have the growth phases, G1 and G2, and we have DNA replication phase, which is S, which means synthesize. And so what these cyclins do, represented by these police officers here, is they regulate this process, making sure that it only goes at a certain speed and that cells do not get too big during their growth phases. And so what happens is, if you mutate these genes, you get rid of these cyclins, and now the cell is going to grow too big, and it's also going to divide too quickly. And that is going to create a massive amount of cells in a short time that are way too big, and that is a tumor. And so that's dangerous. You don't want to mutate your somatic cells randomly. And so what's going to typically happen when you mutate somatic body cells is, at most, you're going to just have an increased risk of cancer. So sorry that was such a long explanation. I'm hoping none of these other questions will be. All right, an important difference between viruses and living cells is what? So to understand this, we have to look at gene expression and understand how viruses sort of hack into this. And so if this right here is a cell membrane, and this is a eukaryotic cell represented by a nucleus, I'm going to say that this is COVID. So what COVID is doing here is it's actually going to find a receptor on your cells, and it's going to bind its receptor to yours, sort of tricking the cell into thinking that this is something good. This is a welcome gift from the rest of the body. And so it accepts in and whatever is inside of this capsule here, and that's going to be a piece of RNA. And so most viruses have their genetic content as RNA, although there are exceptions. There are some viruses that have DNA, and even stranger, there are viruses that have RNA that reverse transcribe their RNA back into DNA, and they can actually integrate their DNA with yours. Those are permanent viral infections. You can't get rid of them. They're things like HIV and things like having herpes. Because once you have DNA integrated into yours, you're going to pass that on to every new daughter cell that you make when you go through mitosis. And so that's going to be permanent. You can't get rid of it unless you do some sort of gene therapy. And so, but the COVID virus, what it's doing is it's inserting what's called mRNA. And you also make mRNA from your own DNA, right? Then your ribosome takes that up and starts taking amino acids from the food that you eat to make new proteins. And so what the virus here is doing is it's hijacking this process. And the reason why is viruses are way, way too small to have things like ribosomes inside of them. And so it cannot make more of itself. It does not have the machinery necessary to reproduce. And so it has to hack into your cell and trick your cells into making more virus. And the way it does that, it inserts its mRNA. And so your ribosome is going to see that, it's going to start reading it, and it's going to make viral protein that will then assemble into more virus. And this will keep happening until your cells basically explode and rupture with millions of more COVID viruses exploding from that. And that can have a lot of bad effects, as we all know, with our current pandemic that we're dealing with. Okay, so again, an important difference between viruses and living cells is that viruses actually cannot reproduce on their own. They need to hack into your cell. And so I really think this is a relevant question for today, and I really wouldn't be surprised if this is the year of COVID. I wouldn't be surprised if you had questions like this on your SOL test. All right, so anyway, that's viruses. Number 9. The eukaryotic organism described above, or I guess below now, would be classified as what? Okay, so we have to have a nucleus, there has to be a cell wall, and chloroplast and mitochondria. So this has to be a plant cell, and so the reason I know that is there's a cell wall. Now, other cells have cell walls, but so the cell walls of plants are made of cellulose, which is a type of sugar, which makes sense because plants make sugar. They can use it for energy, or they can make cell walls with them. And the way they do that is with these things here, which are called chloroplasts, which is one of the things on this list. These chloroplasts are green because they have something called chlorophyll. And it's not stained here, but they also have a nucleus, which is usually a purple stain that does that. You'll notice that these green chloroplasts here are not stained, and that is because they have their own pigment, so we don't actually need to stain them. So the answer here is just plant. Now, if it was a fungus, which I have seen on the SOL, they would look like this, but there would be a little space in between called a septa, and that is because fungi often have septin. They have little openings between them, and the cell wall would be made of something that we call chitin. So if you ever see that, it is a fungus. Number 10. All of these are common shapes except what? So we have rods, sure enough, and that would be here. We call that bacillus. They look like little myconites. Another bacteria shape could be spiral. We call that spirillus. And then we have spherical or round, which is coccyx. And those can all of these can be in a line, and we call that strep. So streptococcus would be a circular bacteria in a line. The only thing that we don't really have is square, because these are single-celled organisms. They're kind of free-floating, swimming around. And I think a square shape would sort of get them stuck on things. And so we don't really find square cells in things that aren't like fungus or plants. We find square cells in fungus, and we find them mostly in plants too, but not in anything else really. Okay, 11. What is the function of enzymes in biological systems? So if you remember chapter 2, enzymes are catalysts. And so what is a catalyst? A catalyst is something that's going to speed up a reaction by reducing what's called activation energy. Activation energy just makes reactions happen faster. Sort of like pouring gasoline on a log before starting the fire. That fire is going to be a lot easier to start with gasoline, which would be a chemical catalyst. Enzymes, however, are protein catalysts, and so they're not used up during the course of a reaction like gasoline would be. Obviously, if you use gasoline on a fire, there's no gas left afterwards, but when you use a protein, there's still a protein afterwards. So that's going to become important in a second. So anyway, what that does is going to reduce activation energy, and you can see that reaction is going to occur much faster. And the way it does that is very physical. There is no magic to it. It's grabbing something like a substrate, and it is manipulating it. Now, the way it might manipulate it is by cutting it, by transporting it, and that's going to help to create a substrate, right? So let's go through these and figure out what the answer is. Does an enzyme act as a product? No, an enzyme is not a product. So just to remember, in a reaction, we have reactants, and those become products. And what an enzyme is, it's right here. Oops. Enzymes are catalysts, which means they go over the arrow. They are not reactants. They're not used up. And they're also not products. They're not the final thing that is made. Right, so it's not actually a product. It's not something that is made. It just assists in the process. Catalysts assist reactions. They are not the beginning of it, and they're not the end of it. They are assisting. And so let's just continue here. So we've got, this is not the answer. Enzymes act as substrates. Enzymes act as substrates. Okay, so a substrate is the thing that it manipulates. Enzymes are not substrates, so we can get rid of that. They manipulate substrates. Enzymes act as catalysts. That's the right answer. Pretty much, if you just remember one thing, enzyme equals catalyst, you will get all of the enzyme questions right. You also have to remember that they reduce activation energy and make things happen faster. All right, so then D, enzymes bond with substrates. Yes, they do, to create the new reaction products. That sort of sounds like, that kind of sounds like the answer, but the thing is enzymes themselves are not products. They help to make products. So I don't think, it kind of sounds like the answer, but I just know that, you know what? I think the SOL test is wrong for making this one of the choices, because D is kind of the answer, but just as a rule of thumb, if you see enzymes or catalysts, that's the answer. So I'm just going to mark off D and say C is the answer. I would not blame you personally. If you put D and I was the person who made this test, I would mark you right. But whatever. Moving on. 12. Many marine invertebrates, like this jellyfish, invertebrate means no backbone, have body surfaces that are permeable. We know what that means. It means things can go through it, right? So, for example, if you have permeable pants and you pee your pants, well, you're going to leak all over the floor, right? Things can move through it. So if they are permeable to water but not salt, osmosis can change the pressure of their body fluids. Fortunately, the ocean is very stable in salt content. What would happen if a jellyfish were placed in a very low salt environment, such as an estuary, which is just a freshwater place? Okay, so here's the thing. Jellyfish live in salty environments, so these little dots are going to be salt, right? And then that means that they must also have salt in their body. And the reason why is water tends to follow salt. Water is a follower. It goes wherever there is salt. So what this means for the jellyfish in its normal habitat is water is going to be coming into the cell to follow that salt, but it's also going to be going out in an equal rate. We call that isotonic. Iso is what you want. That is homeostasis. When you are out of isotonic, you are out of homeostasis. And so, for example, if we took this naturally marine animal full of salt, and then we put that into the wrong environment, like a freshwater pond or I can't think. Your goldfish bowl. What do you call that? Aquarium. I couldn't think for a second. If you put that in your little aquarium full of fresh water, now this organism is full of salt and there's very little salt in the fresh water. So what's going to happen is, again, water will go to where there is more salt concentration, which means water is just going to rush into this poor jellyfish and it's going to end up rupturing its organs and its cells. And that's going to kill the jellyfish pretty quickly, actually. And so the answer here is it would gain water from its environment because it is in a hypotonic solution. So I just remember hypo. So O as in it's swelling, right? So just remember that. Hypotonic is freshwater, hyper is saltwater. Okay, using this tool, Anton von Leeuwenhoek was the first to what? So he looked at microscopic organisms. That's just something you have to know. He's just one of those scientists that you have to memorize. And I'll try to put a list of scientists that you need to remember in this video, and you should look into each one of those. So anyway, he looked at, a lot of people say pond scum, some people say he looked at the bacteria in his teeth. Anyway, it doesn't matter. You just have to remember that he used this sort of primitive adjustable microscope. 14. Using a microscope, a series of cells are observed that lack membrane-bound organelles. And so what are they? So this is a eukaryotic cell, and as you can see, it's very big. It has rooms for nucleus and a mitochondria and all sorts of organelles. Then you look at bacteria, which are much smaller. They don't have that room. Hello, dogs. It's just me. You can relax. They don't have room for those organelles. So, for example, there's no nucleus at all. They are too small. They're about the size of a mitochondria because if you remember, mitochondria entered an agreement with our cells called endosymbiosis. We basically ate them, but we didn't digest them, and they just became our partners. Mitochondria make ATP for us, and in exchange, we give them a nice, safe place to live, and we feed them. And so it's a nice partnership. And so they're about the size of bacteria because they are, or at least were, bacteria. And then if we look even smaller than bacteria, we have viruses. And so viruses are the smallest. They're not actually considered alive because they don't abide by the cell theory. They're not classified as cells and therefore cannot be alive. And that is because they can't make their own proteins. They actually have to hack into other cells like bacteria or human cells in the case of coronavirus and use our ribosomes because they're just too small to even have ribosomes. But anyway, those are all the different kind of cell-like things that we see. And the one that is going to lack the membrane-bound organelles is going to be the prokaryotic cell, of which you should remember there are two. There's bacteria, which, God, I can't write. Let me try that again. There's bacteria. And there is archaea, sometimes called archaebacteria. And it's important to remember that prokaryote is not an actual taxonomic grouping. It is not a monophyletic group, which means that the two members of it aren't actually related. In fact, archaebacteria, although prokaryotic, are less related to bacteria than they are eukaryotic cells like yours. DNA analysis confirms that they are very unrelated to them, despite having a lot of similarity in the way that they look. And they, if you remember, archaea are also called extremophiles, meaning that they love to live in extreme environments like volcano vents and ice and weird stuff like that. Okay, 15, making good time here. In eukaryotic cells, the process indicated by the arrow A occurs where? So in eukaryotes, where do we have DNA? If A is occurring, it must be where DNA is, and that would be in the nucleus, right? So in the nucleus, we are going through a process called transcription. And then we let that RNA out after we have processed it a little bit. Specifically, we put a cap and we put a poly A tail on the mRNA before we let it onto the, into the cytoplasm, where a ribosome is going to come in and do step B. Okay, so that's the nucleus. 16, the weakness of hydrogen bonds between the bases of DNA allow what? Okay, so first of all, let's review what polar means, hydrogen bond means. Hydrogen bonding is between two polar molecules, and what that means is between those molecules, each molecule has electrons that are moving around that molecule, like this water, for example, but those electrons are not moving around evenly. Some parts of that molecule are capable of hoarding those electrons because they're just bigger. So for example, in this water molecule, that red oxygen there is just so big that the electrons are being sort of collected in one part. And what this does is it creates a partially negative charge because those electrons are negative, and then the hydrogen, who has its electrons temporarily stolen, is going to be partially positive. And of course, a partial negative and a partial positive can briefly connect together. This happens in what's called a dipole moment, where there is a positive and negative pole on either end of the water molecule. This is a temporary bond because that dipole moment is just a moment, and that moment will pass. And so this is a weak bond, and we see those bonds in DNA as well because DNA nitrogen bases are also polar. The electrons in them are moving around and being hoarded to one side or the other. And so here we see two hydrogen bonds between adenine and thymine, and here we have I can't remember which is which, but we have cytosine and guanine. Let's see. I'm probably getting this wrong, but I'm going to say this. And they have 3 hydrogen bonds. And so it's just important to remember this base pairing rule. This is called Chargaff's rule. Chargaff was the guy who realized that in all samples of DNA, A is equal to T and C is always equal to G, and that is just because they connect. And so the reason why this is so great is because you can divide this easily. Just like Velcro is weak individually but strong overall, you can start unzipping this because of how weak those bonds are. And so, for example, in DNA replication, we can take advantage of that weakness to break that apart pretty easily, and then we can start creating a new strand. Likewise, when making RNA, we have to open up that DNA in certain places, And we have to start copying one of the two strands to make that RNA. And so that sounds a lot to me like A. Separate during transcription and replication. And so this is replication and this is transcription. So there you go. Long explanation, but that's biology for you. Okay, medicines are being produced using recombinant DNA. Technology. For veterinarians, the use of this DNA technology will result in what? So what does recombinant mean? It simply means if we have a piece of DNA, we can actually take foreign DNA and we can just insert that in there. So for example, if I have a pit bull and I have a Labrador and the pit bull has immunity to Lyme's disease, I could give that immunity to the Labrador if I just insert that pit bull DNA. We actually do this with humans. There's been 3-person children in Russia where we inserted blue-eyed DNA into a brown-eyed child and we gave him blue eyes. That's a real thing. We're living in the future. It's terrifying. But what this is going to do for veterinarians is it's just going to make more treatments for pets. I mean, that's what vets do, so it's a pretty obvious question, I think. Okay, moving on to 18. The arrow in the diagram represents the movement of which molecule? So it's very simple. This is gene expression. We have DNA that we inherit from our parents. It's like a big recipe book. From that, we pull a page out of the recipe book, and that's RNA. And then we read that recipe to make some sort of protein. So for example, melanin is a skin pigment protein, and that creates a trait, like for example, your skin color. And so if you want, I have a video here. I'm not going to play it now just because this review is taking a long time, but I will include it in this Edpuzzle and you can watch it if you'd like. But that process going from the, the flow of information here is called gene expression, and it consists of 2 parts. There's transcription and there is translation. Transcription is this first part here where we're taking DNA and turning it into RNA, and then RNA to protein is called translation. And so what's happening here is DNA is flowing out of the nucleus as mRNA. That M right there means message, and so you can kind of just think of it as a message from the nucleus to go ahead and make something. All right, number 19. Which feature do viruses have in common with cells? So we already talked about how viruses basically have to hack into another cell and use their ribosomes. In some cases, they also use our DNA replication machinery too. And the reason they need to do this is they just don't have room inside of their very, very, very small forms to make this stuff themselves. And so they have to hack into our system and basically take advantage of our machinery. But they do have to have genetic information. So the answer here is, presence of genetic code. So they do have a genetic code. They're just injecting it into us, right? So we already talked about, in the last question, we talked about the veterinarians. We talked about how we have transgenics. That's basically what viruses have been doing since the dawn of life themselves. Okay, 20. Which of the following molecules is most abundant in cells? This is just something you have to know, and you probably already did. It is water. Most of us is water. Our cytoplasm is mostly water. And then we also, of course, have blood running through us, and that's mostly made of water. And the other stuff in us is protein, which makes our cells, and then we have fat and minerals and stuff like that. So there you have it. I'm sorry if that took a while. This just was a very involved section, and I hope it all makes sense to you. I will see you in the next one.